Hi, my name is Ashley Turner. I'm a PhD candidate at the Department of Genetics, and this CAR project is part of the CERTL training that I've been receiving, and this will be completing my CERTL Scholar, which is exciting. The impact of physical 3D models as a supplementary instructional tool on student learning and perception of molecular genetics concepts. Ironically, there are a lot of literature looking at the impact of 3D models on student learning. However, looking at the literature, there's a huge gap in looking at biological sciences. All of the money and resources and studies have looked at human anatomy, the impact of models on human anatomy for pre-nursing. So there's been very little research into the impact of models on just basic molecular sciences, and that was the question we were wanting to look at with undergrad research. Next slide, please. So the questions that we wanted to look at, very simple. Basically, if you put a model in a student's hands, is it better? Do they get more out of it, a more meaningful impact of understanding that biological process as compared to the traditional 2D figure or lecture? So the two questions that we asked were, how do models and model manipulation supplement student learning of those complex biological processes? And what are the students' attitudes and beliefs about the effectiveness of those models? And so I didn't want to list all of the learning outcomes, so I just put it central dogma. But basically, we were looking at DNA to RNA to protein. What's the structure of DNA? What's the process of DNA replication? How is that DNA then taken and used to make the RNA, which is the messenger that leads to protein being produced in a functional product? So that was the goal. That's the model. Those are the model focuses. This course took place in a freshman class. It was a course-based undergraduate research experience, so it was a freshman cure. And I conducted this across 2 semesters with the same, I was actually serving as a TA, and then it was under the same professor. Next slide, please. Looking at the product and design of the methods, we did adapt a tool, so we called it the Central Dogma Assessment Tool, but it was basically adapted and pulled from the CDCI and the MDCA, which is, if you can get information on these, but these are published tools, the Central Dogma Concept Inventory and the Molecular Biology Capstone Assessment. We just took those assessments basically and modified it for our class to teach molecular genetics. There were some components that we weren't going to cover with the models, but you can see the students are actually working with them in these photos. The models takes it through the students actually replicating the DNA and then making their own mRNA. This assessment tool, though, had a narrative stem, and it was followed by 4 true-false statements. There were 4 sections. Students looked at DNA structure, DNA replication, transcription, and translation. All 4 of these kits are available through the 3D Molecular Designs. It's a company basically focused on making biology more accessible and I want to say just easier to grasp, because we all talk about what's inside of a cell, but a person who is having a hard time figuring out what's inside of a cell, this is a model that you can wrap your hands and your mind around. So next slide. It was a mixed methods design, so we looked a little bit, as mentioned earlier, the most popular is pre, post, and a final assessment. We did do a final assessment, so the pre was directly at the beginning of the class. It was followed by the actual model itself, so the first setup was the DNA models, which is in that purple triangle, and then we did another pre-assessment, so split the assessment into 2, had them focused on the 2 areas, and then they did a post-assessment directly after 10 days. And then at the end of the semester asked the question, was it just something short-term or was it long-lived? Did they actually gain something? And then we also looked at a Likert-type survey, and then we had them answer questions. They were also able to provide free response on what they felt were their misconceptions. I am showing you, though, this semester, we did do it 2 semesters. I'm still churning through the data. It's not large ends. It's just a kind of a complex data set with as many things as we had with the mixed methods. So what I'm showing you today is just a preview of the 16 for spring, but I can tell you preliminary that it is holding true to be the same trends and conclusions. Next slide. So if you can look at this slide, this is just basically looking at the impact of those models. So you saw the students, they had the DNA in their hands, they're adding the nucleotides, they're trying to figure out what enzyme opens up the DNA. They're actually walking through the process of those biological concepts using a model. And so what you can see, you're looking on the left in that table, is I did a simple, just a paired t-test from pre to post, post to final, and pre to final. And what you can see is looking at pre to post, the students had a significant gain in scores on that assessment, and they also had a significant gain at pre to final. So it wasn't something that was just present from pre to post in that short term. It was also not as significant, but it was still improved and significant as compared to the post. There was no difference between post and final. looking at the spread of students and their assessments on the test, which shows that if they got it from the models during that short term, then they didn't gain anything in the long term of that they picked up that was not from the models. What's shown on the right is just a different score. It's showing the scales. What you can see is that there is an increase in terms of looking at the number of points from 0 to 6 from pre to post. The blue shifts to the right. There is a little drop off in the red. Students do have, reinforcement is important, and so you have to keep reinforcing some concepts, but overall they still do better on the assessment, even in long term at the final of the course. Next slide. Students also had a very positive perception. So this is just a graph showing, this is the Likert type scale, but we had a number of different STEMs, and they could put their opinion, if they agreed, strongly agreed, neutral, disagreed. What you can see is there's a lot of red and blue, and those are the strongly agree and agree, meaning those models for students made them the concepts easier to understand. It also made them a little bit more learning and engaging, but as you can see, there are still some students who say that they made more concepts more confusing, and there were some students who still said that they would much rather have the traditional method. So overall, largely positive student perception, but there are still some students who still prefer other methods of learning as compared to the models. And I love these student misconceptions. Only one replication bubble was made, is what they thought. It was only at the one replication bubble at the one point on the huge DNA. They also thought, one student quoted, I thought it was quick and foolproof. Turns out proofreading is important. They were making their own DNA and making mistakes. So the process of them actually doing it showed them the importance of the machinery and all of the components for those complex processes. Next slide, please.
Okay, you're at 7 minutes.
Yes, yeah, I'll finish here. The implications for teaching are just general. We're still learning from this, but the 3 models overall increase student learning. Repeated assessments were key for us in looking at short-term and long-term gains. And where we're wanting to really go with this is the intervention method, looking at those misconceptions early on, and at that short window, targeting that misconception, and then at the end of the semester, seeing if that targeted approach made it better. Also, obviously, being able to see a student that has mastered something completely and you're trying to offer them an enrichment opportunity. Student perception may not be the best tool to assess learning. There were students who loved the models and they didn't have as large of gains as if you look at an individual by individual basis, but a student-free response might offer more insight into learning, and that's been seen over and over again. And I just go back to repeat, one size don't fit all. One size doesn't fit all, and this applies still in terms of the 3D models. Differentiated learning still should be the goal in the curriculum, even when looking at something so novel as interactive learning or a 3D model setup. And I said that's it. You can go to the next slide. And my acknowledgements are my mentor, my teaching and research mentor, Anil Chawla at UAB, and the CERTL program at UAB, and I'll take questions.
Great. So now we do have time for questions. It looks like Shauna has already written a question in the chat. Did you compare the learning score changes versus lecturing without the models? And could you compare the final exam scores to previous years?
So what for me this was new for myself and my teaching and research mentor. So I don't have any data from like the traditional lecturing style setup where we were mentioning like a control class where they just received the traditional quote unquote lecture format or textbook figure. So I don't have that to compare to, but I guess what I was hoping the students to be their own internal control for pre and post, but I don't have the comparison of the regular class.
Great.
What are your thoughts on the students that said the models can make certain? Oh yeah, okay. So some of the students actually, what you notice is that the students were working in groups. You can see a lot of the students were working on the groups together. What I found, and this is going to be part of the data, but the students who I felt the models were confusing were also individuals who did not prefer group work. So if a student didn't like group work, they did not get a lot out of the model because they were not comfortable in a group setting, or they didn't feel comfortable in their group to begin with. They just kind of preferred individual solo work. So next time we want to offer the models to be open for students to be able to come back in as individually and kind of make sense of the understanding. And then, have you considered using data from other previous classes for control? Yeah, and that's a good question. I'm going to reach out to a regular biology course and see if I can get someone to have their class take possibly this assessment pre and post a particular, just a one lesson or a couple lessons over the same topic and see if we can get some data to compare to.
Great, thank you.